So for those of you I haven't met, my name is Stephen Bagley. I'm a neuro-oncologist and clinical trialist and clinical researcher at the University of Pennsylvania. And in addition to therapeutic clinical trials, I've done a lot of work in the liquid biopsy space for primary malignant brain tumors. And so I want to just spend a little time trying to get everybody on the same page for today's discussion in terms of the current state of the field. The only relevant disclosure I have is that I have worked with C2I Genomics in Tempus on a research basis, and I will discuss some of that data. So I want to try to convey very clearly how much of an urgent and unmet clinical need effective liquid biopsy is for brain cancer. And I'm going to do my best to make this both comprehensive and brief in terms of a current state of the field. And then lastly, share what I view as key future directions. So just to give you a sense of what we're dealing with here, this is primary brain tumors at a glance. And by far the most common in adults is glioma. And you can see that within that, the major slice of this pie here is glioblastoma, which is a grade 4 glioma. There are over 16,000 new gliomas each year in the United States alone. The vast majority of these, again, are glioblastoma. And I'm really going to focus for the rest of this talk on adult glioblastoma as our motivating example, recognizing that many of these issues I'm going to discuss apply to both other primary brain tumors as well as the pediatric population. So for those of you who are not terribly familiar with this disease, this is a diffuse infiltrative tumor that arises from neuroglial stem or progenitor cells. This is what it looks like on imaging. And on pathology, we know that this is actually a disease of the diffuse brain. Even though you may have a discrete localized tumor shown here in the gray in this image, these little black dots actually represent areas where there's microscopic tumor infiltration. So you can see that this disease infiltrates along white matter tracts, and from the very get-go is really a diffuse process. And in part because of this and a variety of other issues, this remains a near uniformly fatal disease. And this is despite an aggressive and somewhat toxic standard of care therapy that consists of radiation and temozolomide chemotherapy, the core components of which have remained essentially unchanged since 2005. We've had tumor treating fields that have further improved survival, but we're still looking at, in general, at least in the real world population, median overall survivals of 12 to 15 months. And so, it goes without saying that if you ask a patient or their treating physician what they need, we need more effective therapies. There's no doubt about that. But shortly behind on that list would be more accurate and precise disease monitoring tools, which I'm going to get into, and less invasive diagnostics. And I would argue strongly that if we're able to do numbers 2 and 3 better, it will in turn lead to more effective therapies, really in the realm of clinical trials. So chemoradiotherapy, standard of care for glioblastoma, commonly produces pseudoprogression, which is loosely defined as a transient worsening of contrast enhancement on MRI. And this is essentially due to breakdown of the blood-brain barrier from radiation. And as you can see in this typical case shown here, this actually resolves on its own without a change in treatment in some patients. However, this looks just like tumor progression. And so while the term pseudoprogression has really caught on in recent years in oncology in the setting of immunotherapy, this has been a problem for glioblastoma patients long before the era of immunotherapy, and has actually only been further compounded as we're starting to evaluate some of these immune-based treatments in clinical trials. And so this makes it very, very hard to take care of these patients. One can imagine that if there's an effective liquid biopsy metric, something that we can use to actually monitor this disease, it could be used to augment MRI and make both clinical trial and standard practice management easier for these patients. The other area that there's potential for liquid biopsy is really for non-invasive diagnosis and next generation sequencing. And so the typical course for our patients is that because their brain mass is often large and symptomatic, we need to resect it anyway for therapeutic reasons. So we end up with tissue that we sequence and then informs both prognosis and in some cases therapy. But there are other cases, for example, here, a brainstem lesion, where this is actually not a trivial task. Even a limited biopsy carries significant risk. And we would like to know, of course, the same genetic features we'd want for any patient in those cases. And the other area that's really important is for, cases where, we actually could use CSF or blood to try to get the same information that we would get from tissue sequencing. There's also patients who may have a recurrence distant from their original resection. So this is a case where, you can see the original resection cavity here on the left, and then there's a contralateral recurrence that's non-operable. And we know that with glioblastoma in particular, the molecular profile at recurrence is often quite different from what we see at the initial diagnosis. So getting updated molecular information here through CSF or blood would spare that patient an invasive and potentially morbid brain surgery. So to summarize this, we can establish diagnosis in cases where surgery is either impossible or high risk. We could avoid repeat surgeries to obtain up-to-date tumor genetics at the time of recurrence. And we can detect potentially targetable genetic alterations that might actually be missed by concomitant tissue sequencing. And this speaks to the marked tumor heterogeneity that is characteristic of glioblastoma. So in terms of an overview of what's been looked at in the literature, cell-free DNA, both in the plasma and CSF, has really been the abundance of literature in the neuro-oncology field, just like it has been across oncology. And this is where I'm going to focus the remainder of the presentation. But it is worth noting that both EVs and CTCs have also been studied. It's just that the studies have been much smaller. The methods for isolation have varied widely in the field. And so this is an area that I think is really promising, but is still in extremely early days. And I'm not going to revisit this for the remainder of this particular talk. So the main problem so far for this field has been the blood-brain barrier. And we know very firmly at this point that traditional liquid biopsy, and by that I mean sending plasma for a targeted NGS panel, really has limited clinical utility. Chetan Bedagowda and colleagues showed this nicely in their landmark paper in 2014. This is just the rate of detection of any ctDNA in the blood, a variety of different tumors on the x-axis. You can see gliomas all the way over here on the right. Very few patients have detectable alterations. This was a study we published a couple of years ago looking at matched plasma and tissue in the newly diagnosed GBM setting. And the color coding just shows you which mutations were found, in tissue versus plasma versus both. And the key takeaway is that there were essentially no mutations detected in both. The green boxes were actually variants in the same gene, but different variants. And so this is really what we get when we just try to send plasma for targeted NGS in this disease. What is potentially more interesting, I think, in an area that both myself and others in the field have really focused our recent efforts has been on tumor-guided sequencing. Here, you're not able to, de novo detect mutations that that doesn't supplant tissue sequencing. But if you know what you're looking for based on tissue sequencing, it's easier to find in fluid compartments. And so this has real utility for disease monitoring and response assessment. There have been a couple of really nice examples in the literature, one using personalized hybrid-based capture for multiple mutations of interest. So Moulier and colleagues showed this in a nice paper. They actually did multi-region tumor tissue whole exome for each patient in this cohort. And then they used those mutations detected in tissue to generate targeted panels. And then they did high-def sequencing of CSF, plasma, and urine. You can see each of these patients on the x-axis actually had a fairly high cfDNA fraction when detected in CSF, plasma, and urine. And then the other area is if you have one particular mutation of interest, and a good example for glioma would be TERT promoter mutation, since that is a very common alteration in this disease. This was a multi-center study published in CCR a couple years ago that looked at TERT promoter droplet digital PCR. And you can see again here, really nice signal for the TERT mutant patients. This is in the plasma. Whereas you don't get the same signal in TERT wild type patients, healthy controls, or other CNS diseases. What we've been doing in collaboration with Asaf and the C2I team has actually been to look at whole genome sequencing of plasma under the same sort of idea. And what we did here was hypothesize that since there's so little ctDNA in the plasma of these patients, if we perform whole genome sequencing of the tumor tissue first, that may allow for generation of personalized tumor signatures that we can then detect in the plasma. So again, a form of tumor-guided sequencing. And so using their proprietary C2I intelligence platform, we actually did a pilot study with them. And again, the overarching theme is that the breadth of sequencing with whole genome will supplant its limited depth of sequencing to overcome this barrier of very low ctDNA levels. So for this initial pilot, we looked at 37 patients with newly diagnosed GBM with matched tumor and plasma samples. The tumor tissue is subjected to whole genome, and then C2I's algorithm integrates the single nucleotide variant and copy number variant calls to generate this personalized tumor signature. We then perform whole genome on the plasma cfDNA, to try to detect that signature. And the readout is tumor fraction, which we were able to generate at a variety of time points shown here in this schema, which reflects the usual clinical course for patients with glioblastoma. And so these are just two examples from this cohort of really nice correlation between the copy number variation profiles and tumor tissue and plasma. This patient had gain of 7, loss of 10, a very common copy number variant profile in GBM. We see the same thing in the plasma. And then in patient B, gain of 7, gain of 18, same thing in the plasma. So suggesting that this is actually looking fairly promising. So the first step we wanted to kind of do here was see if there was any correlation with survival. So what we found was that at the first post-surgical time point, so this is actually post-op day number 1, if you draw plasma on these patients and measure the tumor fraction in this way, there's actually a striking difference in overall survival between those patients with high tumor fraction, shown by the red line here, and patients with low tumor fraction, shown by the blue line. This came out to a median overall survival difference of 8 versus 19 months. And this association persisted after we adjusted for all the other relevant prognostic factors in this disease. And so it raises a very interesting question, which is, could this potentially be used to assess an MRD state? And the term minimal residual disease is complicated when you're talking about a disease like glioblastoma, where we never really get to 0 or that close to 0. But it could be very useful as a risk stratification tool for understanding which patients are high risk from the very get-go. So more on this to come, I hope, in the near future. I do want to highlight a couple of other areas that have been, I think, really interesting and showing significant promise in this field. So Prasad Nassiri and colleagues published a cfDNA methylome paper a couple of years ago in Nature Medicine. And this was using plasma cfDNA methylation profiles to actually discriminate between a variety of primary intracranial tumors. So they use cell-free methylated DNA immunoprecipitation and high throughput sequencing, or CF-methyl dipseq. And for this initial study, they looked at 447 plasma samples across 9 tumor types that included 60 patients with glioma. And the binomial random forest classifiers, glioma versus all the other cancers they looked at, were trained using the top 300 differentially methylated regions for glioma versus other. And this is what it looks like. You can see all the way on the right, all the black bars are the glioma patients with a markedly different cfDNA methylation signal compared to the other tumors. And when you try to actually discriminate between various intracranial neoplasms, it does so with fairly decent accuracy. So these are the AUC numbers for IDH mutant glioma versus other, low-grade glial neuronal tumors versus other, and lastly, IDH wild-type glioma versus other. So I think this is an interesting area to look at for non-invasive detection and discrimination between the CNS tumors where surgery is either too difficult or too risky to perform. And then I want to spend just the last little bit of time here focusing on cerebral spinal fluid. So this clearly has more intimate contact with the tumor than plasma. And we know pretty firmly at this point that the concentration of ctDNA is much higher compared to plasma. And what's also nice is that the CSF is paucicellular, so you get less background signal when you're trying to detect the ctDNA and sequence it. So I think that one of the kind of largest papers looking at this came from Memorial Sloan Kettering a couple of years ago. Alexandra Miller and colleagues looked at 85 adult patients with glioma, and they were able to detect at least one mutation in the CSF of about half of the patients. And this figure here shows you the 30 CSF and tumor matched pairs that they looked at, and gives you a sense of how many mutations were shared between tissue and CSF, how many were tissue only, and how many were CSF only. And you can see that there's actually a lot of variance in terms of shared mutations versus exclusive to one compartment. And so this was I think a really nice first look at this, but also sort of raised a lot of questions. I think these are really important unanswered questions for CSF liquid biopsy. Number one, does tumor location matter? There is some data to suggest that, and I put a couple of the references at the bottom here, that if the tumor is closer to a CSF reservoir, you get higher yield of ctDNA rather than being fully encapsulated by brain parenchyma. How do the results differ when CSF is obtained via lumbar puncture versus if you actually obtain it through cisternal puncture intraoperatively? And we have some unpublished data suggest it varies a lot. And then how do results differ when the CSF is fresh versus frozen and then thawed and sequenced? And is the information obtained clinically meaningful enough to really warrant going through what I'll call a semi-invasive procedure in lumbar puncture? these are important questions moving forward. So I will end by asking, why now? Why should we care about this at this moment? So, this is a summary of the current GBM clinical trial landscape that I published as part of a think tank through the Society for Neuro-Oncology last year. And this just gives you a sense of these were the 136 currently accruing GBM clinical trials at the time in the US. And there's a wide variety of immunotherapy, targeted therapy trials, and even a good number looking at combinations of all of these. And so we know that with this increasing number of immunotherapy trials, the ability to discriminate between progression and pseudoprogression becomes critically important. And if we can't figure out how to do this better, there's a lot of studies that are either going to generate false signals, or we may actually miss some therapies that truly are effective, but patients seem to be blown through with pseudoprogression. And then there's an increasing number of molecularly targeted therapies, and the results of NGS testing actually may inform eligibility. I'm pasting here one example of the trial that I'm leading at Penn. That's looking at dual targeted or bivalent CAR T cells for EGFR amplified recurrent GBM. And here we mandate that the tumor is known to have and maintain EGFR amplification at recurrence. And so you can imagine for a patient with a non-operable recurrence, it would be great to be able to do plasma or CSF sequencing to confirm that alteration, that target is still there. And then lastly, I think the momentum in the field is really accelerating right now. There's a global appetite for really working together to move this field forward. Patrick Wen is one of our panelists today, leads the RANO group, and they recently published a paper shown here in Neuro-Oncology that is really kind of laying out a roadmap for where this field should be going. So in summary, liquid biopsy technologies, I think, hold great promise to improve the care of our patients with primary malignant brain tumors, including GBM. And this could be both in the realm of non-invasive diagnostics and molecular profiling, which I think would enhance both standard therapy and clinical trials, and then also improved disease monitoring. And if we can do this better, we can detect progression earlier and more accurately. And that benefits patients both in the trial setting and in standard practice. The major areas I highlighted include tumor-guided plasma ctDNA sequencing, cfDNA methylomics, and CSF ctDNA sequencing. And as I think as a field, and this is what I'm excited to talk about today with everyone, is really, how do we think about standardization for biofluid collection, processing, storage? How do we choose the highest priority analytes, and what's the best way to detect them? And then I think above all else, we need to work together to do prospective, multi-center, and most importantly, adequately powered studies. If we continue to do small, single-center, kind of approaches, we're not really going to be able to move this field forward in a meaningful way. So this is my very last slide. I'm going to show what I hope will look like the not too distant future. So you can imagine a patient shows up with headaches, gets a scan. The neuroradiologist tells us that this is either glioblastoma, but also has some features that are consistent with CNS lymphoma. And for those of you who don't take care of these diseases, glioblastoma, we typically want to do a maximal surgical resection, whereas for CNS lymphoma, surgery is not beneficial at all. And we actually want to proceed right with systemic chemotherapy. So we could perform cfDNA methylation profiling and actually make the diagnosis. And in this case, let's say it does show glioblastoma, we go ahead and proceed with maximal safe resection. And once that tumor is confirmed, we're actually able to do the whole genome sequencing I had mentioned, both in the tumor tissue and the plasma. And the patient's followed over time with both serial MRI and serial blood draws. We ultimately will get that radiographic progression, not knowing if it's real or if it's treatment-induced pseudoprogression. So we check a blood draw, perhaps it's some other assay looking at ctDNA levels. We see the signal rising, we're concerned this is actually progression, and we go ahead and evaluate the patient for trials. In order to be eligible for this particular trial, an EGFR targeted trial, we don't necessarily want to put this patient through another brain procedure. So we perform lumbar puncture, sequence the CSF, find that it does indeed maintain EGFR amplification, and we proceed with our clinical trial. So this is where I hope we're heading over the next few years. Thank you very much for your attention. I want to acknowledge Erica Carpenter, who's a key collaborator of mine and has helped me to generate a lot of the data I showed, and then also Asaf for his help with our C2I genomics project. My email is here if there's any questions or follow-up. Thank you so much.